So we have learned the basal side of a cell, of an epithelial cell, and we have learned what's present at the lateral sides of the cells. But what we haven't studied are what's the apical modifications or apical surface of the cell and what it contains, right? So there's some apical modifications that are in your syllabus that I will mention briefly here, and then I will explain to you those. So firstly, we have the microvilli. Yeah, this. Okay, so microvilli. These are little projections that help increase surface area for absorption of nutrients and food. Then we have number 2. Okay, we have cilia. Yeah, and I think you guys should have an idea of what the cilia is. The cilia is a motile hair-like particle or apical modification that the cells have, and these are able to move and circulate stuff that's present outside the apical surface of an epithelium. And thirdly, we have what's called Stereocilia. This might be a new term for you. These are cilia-like modifications, again hair-like modifications, and these are actually present typically in the testes and the inner ear. So for illustration, I'll draw like an ear cell here and maybe draw some stereocilia here. You'll understand how they function in the ear chapter that I will do, the special senses chapter, at the end of this series. I think it's the last chapter of the series. And so let's go ahead and try to look at this diagram, which actually contained the microvilli. So here's some microvilli on this intestinal columnar epithelial diagram of a cell, and the microvilli are obviously apical projections that increase the surface area of the cell, and they are actually made up of actin, so there would be lots and lots of actin underneath over here, and they are basically protrusions of the cell surface membrane to increase surface area. Yeah, and when you increase surface area to volume ratio, there's a greater rate of absorption of nutrients, which is very, very welcomed by intestinal epithelial cells whose function is to absorb nutrients. So what the microvilli usually assist with is absorption of stuff, right? For example, nutrients and food and water. Food, water, absorption. Next up we have, before moving on, I actually have a microscopic slide that I want to show you. So this is the first microscopic slide of the series. Congratulations guys, you made it here. Okay. So as you can see here that the individual microvilli is actually not visible on a microscopic slide, right? It is that small, it is that tiny, but those little tiny projections actually help increase a lot of surface area for that absorption of nutrients and so on. So if the individual microvilli is not visible, then what is seen? That's the question that arises, right? And what is seen is actually a very thin border on top of these cells, on the apical surface of these cells, as you can see here. So you see this border right here? This is called the striated or brush border, right? Let's call it brush, as it looks like a brush, right? You ever seen a brush which has many different strands hanging from it? Just like the microvilli hang from the apical surface of a cell. This is also called a brush border, also called a striated border, and so on. But these are actually just lots and lots of microvilli that appear like a very thin border, which we call a brush border in histology. So what you'd assume is obviously that microvilli cannot be seen by a microscope. Now, there's a clinical correlate to this, and that clinical correlate is called celiac disease. And I think some of you guys might have an idea what the celiac disease is or what it relates to. It's actually a disease which results from gluten sensitivity. Gluten sensitivity. So if a person who is sensitive to gluten, if he eats a gluten-rich diet, then what happens is that the microvilli of his intestinal epithelium would be damaged, right? And when there is a loss of microvilli, there is a loss of what border? Brush border or striated border, right? So brush border will be gone, striated border will be gone if a person having gluten sensitivity, if a person having celiac disease eats a gluten-rich diet. What are some of the gluten-rich things? Could you tell me? Well, all kinds of bread, right? Unless it's obviously labeled gluten-free bread, all types of bread made up of wheat, yeah? Even beer. If you like drinking beer, beer is usually full of gluten as well. It's gluten rich. So the brush border will be lost and there would be malabsorption. Because you can't absorb the food anymore. We've lost the brush border. So malabsorption. And malabsorption obviously would lead to deficiency in many different things, many different vitamins and nutrients and so on, and cause many pathologies, many different symptoms, many pathologies. Any further pathologies and so on. Okay, next up we have the cilia. I'm not going to waste too much time here. I'm just going to start with the next apical modification. So this is a histology slide, microscopic slide of the respiratory epithelium. Do you know what the respiratory epithelium is? The respiratory epithelium is present in the respiratory tract, like the nasal passage and the trachea and the bronchi, right? So the respiratory epithelium is actually a pseudo-stratified columnar epithelium. I'll tell you what that is in just a bit. But they have these columnar cells, and on their apical surface they have these modifications that are called cilia. And we said in the introduction that cilia they're motile and they're able to move using ATP and able to circulate the mucus that will be produced in this respiratory epithelium. And actually we have labeled right here the goblet cell. And goblet cells are actually the cells that are responsible for producing that specific mucus. So goblet cells would produce the mucus here, and then that mucus would be basically brushed off upwards in our respiratory tract by these cilia. So I'm going to mention here, even though I will mention it later as well, the cilia is present in the respiratory tract or respiratory epithelium. And what is cilia made up of? Well, it's actually made up of what's called axonemes. And these axonemes go ahead and form axonemal dian, which performs the function of motion. Now the last one we have is Stereocilia, which I'm not going to go into too much detail here because it has different functions. The function that it serves in the inner ear hair cells, or you'd say hair cells of the ear, hair cells of the ear. The inner ear has some hair cells, which are basically called hair cells because they have these stereocilia, and they are sensitive to vibration. So if there's any vibration coming from the surrounding of that person, of any person, you hearing me right now, you have these stereocilia that are actually moving because of the vibrations of my voice, and the vibrations are actually causing flow of ions across the membrane and so on. The mechanism of this, I will explain it later on in the chapter. Even though it's concerned with physiology, I will still explain it just for the sake of learning. And then another place that they're present in is the epididymis. And yeah, that's it for this video. So I'm going to go ahead and try to see the neat notes of what we've got. Actually, here's some examples of the hair cell. If you can see a diagram of this, or you want to see a diagram of this is the hair cell of an inner ear. So here's the apical cell surface specializations or modifications. We have number 1, microvilli, then the stereocilia and the cilia. And next video is going to be about the types of epithelia. Alright, I'll see you guys in the next one.